EPPO-Q-bank is composed of 7 databases, one for arthropods, one for bacteria, one for fungi, one for nematodes, one for phytoplasma, one for plants, and one for virus and viroids. Each database are organized the same way. You first have a home page with details about the specific databases. On the left, you can see the statistics for this database, so the number of species, specimens, and sequences in the database. You also have information about the curators of the database. Then you have one page that lists all the species included in the database. You can search species by EPPO code, preferred name, and EPPO categorization. You can also use this box to filter the species by either scientific names or EPPO code. You can also see here in these two columns the number of specimens and the number of sequences that are present in the database for those species. So for example, if you're interested by Anoplophora, you can see here that we have 2 species of Anoplophora, Anoplophora chinensis and Anoplophora glabripenis. You can see the number of specimens for each of those species and the number of sequences. If you click on one species, you will go to the species specific page. You first have an overview of the species, which comprise information from EPPO Global Database with links to EPPO Global Database. And you also have a direct link to the specific diagnostic protocol regarding this species, if there is one. And then if you click on specimen, you will have the list of specimens for which sequences are available in the database. You can view each specimen one by one by clicking on View. You will have first the general information on the specimen with the name, and then some metadata regarding this specimen. So for example, the stage of the specimen, origin, the sampling and isolation date, and by whom this specimen has been identified. You will also have information on the collection that holds this specimen. So here it's a specimen that is held by the CBGP Continental Arthropod collection from Inhai. If you want to contact the collection, you can click on Contact, and a contact form will allow you to ask a question about this specific specimen. If you go to DNA sequence, you can see there is one CO1 sequence available. You can click on View to see the sequence. Some statistics on this sequence are also provided. Please note that you have to be connected to be able to see the sequence. If you are not connected, please log in. You can also search for a species or a specimen directly from the main menu using this search bar. The results are displayed in a table, and you can click on the species of interest. In the section Methodology, you can find information on tests available for the identification of quarantine pests. Those usually are barcoding tests published in EPPO PM7129 diagnostic protocol, but other tests may also be described. A molecular scheme for the identification of specific pests using barcoding is provided, as well as a direct link to PM7.1.29.